Good morning, everyone. Belgium, Europe, the world, for an exciting meeting this morning, a special meeting. In total, 4 students of the IMBRSea Master Program of the cohort of 2 years ago, actually, that will present their thesis 2 years ago. That started 2 years ago in the master program. It's not that they were assumed to finish 2 years ago. So 4 students will present their master thesis today via this online meeting. So each student will first present for maximum 20 minutes. And then afterwards, there's time for discussion. As you are present maybe on the live chat as well, on the live video feed, you also have the opportunity to ask questions yourself by just typing in the questions in the chat box that is on the site. And then during discussion, we will also, of course, take these questions into account. So we start with a presentation by Jan Bierwit, who did a thesis project on the restoration of marine sponges, and he will talk about actually what you can learn from an experimental cultivation and see whether these techniques are indeed useful techniques when transplanting sponges to new environments. So Jan, the floor is yours, and good luck with your presentation.
Thank you very much, Tim, and good morning, everyone. Today I'm going to present you my thesis and also the results of my thesis, restoration of marine sponges, what we can learn from over a century of experimental cultivation. First of all, I'm going to talk about the aims and objectives before I give you a brief introduction, and then of course explain my method and materials before I go into my results, discuss the findings, and finally draw my conclusion. So the idea was to provide guidance and recommendation on best transplantation techniques for distinctive groups of sponges, especially in terms of future restoration projects. And sponges have been cultivated already for quite some time by humans. So the idea was first to create a review, to compile information and synthesize previous results, and then also couple these with field experiments that were carried out by the university in Ancona previously already, and I had access to the data, and then to also incorporate findings of both to finally give some guide and recommendations. As I mentioned, first of all, a quick introduction. I think it's very important to know at least a little bit about the history of marine sponge exploitation. And on the left side here on the pictures, we can see sponge depictions on Greek vases, and they've been found even 2000 BC already, and which indicates that sponges have been used very early already, and the sponge populations have been altered by humans through exploitation and harvest at a very early level, mostly because they were used as so-called bath sponges, bath utensils, which you can see in those pictures here. They normally have a very roundish form, and they're very compressible and resilient. And like I said, they've been used as bath utensils, but also, for example, by Roman soldiers to polster their helmets and similar appliances. It mostly started in the Mediterranean region, of course first in the coastal region, Greece for example, Italy, but also in the northern African part, and from there it really made its way into continental Europe mid-century, and Paris became a very big hub for sponge trade, exporting it all across Europe, but even also across European borders. As you can imagine, with heavy exploitation, the sponge populations, especially in the Mediterranean, decreased very heavily, especially certain species, and they even nearly faced local extinction. So throughout the 20th century, the populations decreased very heavily, and of course also the trade of sponges dropped a lot. At the end of the 20th century, there was also the discovery of secondary metabolites in sponges and also other bioactive compounds that are very, very interesting for the pharmaceutical industry. On the graph on the bottom, we can see the natural products derived from different taxa, and actually the dark blue area is Porifera, and we can see it has really seen a huge increase since the 90s. So because of this heavy exploitation, especially of bath sponges very early, and the drastic decline, humans started very early to think about alternative ways to produce and harvest these species. So for more than over 100 years, humans and researchers have tried to cultivate them artificially. And my idea first was to establish a review on the in situ techniques of sponge cultivation. You can see the search procedure in the table on the right side, and also try to classify all the methodologies used, because the information so far has been relatively scattered. And I compiled a very large table, which allowed me in the end for the comparison of survival rates in regards to methodology, materials, taxonomy, and other characteristics. You can see, for example, in the table, this is just a screenshot that I extracted useful information like commercial interest, materials, the species, but also the survival rates, and I added certain sponge features such as the type of skeleton and other useful information. I also mentioned there was an experimental part where different species were transplanted previously already, which were Spongia lamella, which we see on the top. It was transplanted using plastic dowels and epoxy putty. We can see it here, the dowels were inserted into the sponge. Before this happened, they were actually cut into pieces, like we used fragments, and they were left on a vertical rope for acclimatization period to allow the sponge to regenerate after the cutting, and also attached to the plastic dowels. The transplantations were already conducted in 2016 for Axinella polypoides. Two different methodologies were used. They were either transplanted vertically in like an erect position, which you can see on the left side, using the same epoxy putty as for Spongia lamella, but this time directly on the sponge, and also in a more horizontal position, which we can see on the right picture. It is also to say that for the horizontal approach, the fragments were slightly larger than in the vertical approach. Sometimes they're already branching off a little bit. The vertical methodology was used in 2020, and the horizontal method was used in 2021. Then there was also Chondrosia uniformis, which is a very high plasticity and also deformability of its body. So it has been very challenging in the past to cultivate. And actually what was done here is that sponge fragments were dropped on an artificial surface, like huge concrete boulders that were dropped originally to prevent bottom trawling, and they were covered with a PVC grid mesh, which you can see here. And the mesh was then removed after 2 months, approximately, after the sponges have attached naturally to the surface. Of course, all experiments were monitored on a regular basis with photo surveys, which allowed for the calculation of survival rates and also 3D image analyzing to estimate the growth of the surface area and also the volume. So, looking at the reviews, I first highlight the geographical distribution and commercial interest. The distribution we can see on the map on the top over here. The gradient actually indicates the number of papers that have been published, and we can see the dark areas are the hotspots, which is here the temperate northern Atlantic, which also includes the Mediterranean, and the central Indo-Pacific, which also includes the part of the Great Barrier Reef in Australia. The percentages for each ecoregion we can also see on the bottom part here in the pie chart. Most of the studies actually in this review actually focused on bioactive compounds as commercial interest, lesser focused on web sponges, and only very little focused on restoration. We can also see the distribution of the commercial interest for each ecoregion over here in the bar plots, and A stands for a broader general ecological topic. Yeah. Then what was also very interesting in terms of commercial interest was that there was a shift since the early 2000s approximately towards bioactive compounds. Before it was more focused on bath sponges, but with the discovery of especially secondary metabolites and the interest of the pharmaceutical industry, there was really the shift that could be observed. As mentioned, I also tried to classify the different methodologies, and I grouped them into 6 different categories, and 3 of them I used for my analysis. The others I actually excluded because the number of trials was relatively low, in some cases even just 1. So first want to highlight the graph on the top left corner, where we can see the survival rates of the different methodologies. And actually the differences are not very strong, and there was no significant difference found. So what I found very interesting is the graph on the bottom left, where we then actually included also the skeletal consistency. And we can see that within each method, there's actually quite some differences, which is a very interesting result, because before sponges were usually thought to be, sponge cultivation was thought to be very, very species dependent. But it seems like that there is other patterns too, like the skeletal consistency, which could also be paired with the taxonomy, what we see over here, that could inform future restoration projects, for example, which is very useful. And also for the experimental part, we actually had very high survival rates after 2 months for all the species, except Axinella polypoides, transplanted vertically. And this is mostly because the species is very susceptible to touch, and if transplanted in an erect vertical position, it often detaches due to necrosis at the bottom part. So it seems that the horizontal method is more appropriate for this species, although it still has to last over winter, and we have to check the spring, because with the horizontal method, they were only transplanted in 2021. And actually, from the cohort from 2020, all the explants were lost during winter. There was some lost before already, as you can see in the graph, but then during winter, on a longer period, we even lost all of the explants. Then the growth could only be determined for Chondrosa reniformis. As I mentioned, we lost all the Axinella polypoides fragments from the 2020 cohort, and the 2021 cohort was transplanted just before the thesis, so there was no adequate growth yet. And then for Spongia lamella, the amount and the quality of the pictures unfortunately did not allow for adequate growth measures. For Chondrosa reniformis, what was very interesting is that we sometimes observed growth in volume, but not in surface area, and also the other way around. And this is mostly due because it's a very plastic species, like I mentioned before. It can really move around on the surface and change its body, and it has some form of natural contraction and expansion, which we can also see in the graph over here. The dotted lines actually represent the mean change in volume of 2 different groups of Chondrosa reniformis, and we can see this contraction here, how it expands afterwards and then contracts again. And the same was also observed in a lab on a very time scale, even a very short time scale, even over hours. This species really contracted and expanded, so that's very interesting, but also due to its deformity, very challenging in times of cultivation. So then in the end, what did we learn? First of all, I think it's important to mention that marine sponges still are relatively underrepresented in scientific literature compared to other benthic key habitats, for example, coral reefs or seagrass beds, which have seen very high interest, and especially in terms of restoration. When we think of restoration, for many people, of course, coral reefs pop up, even seagrass beds, but marine sponges, they're just starting to be recognized as important key species. Then, as I mentioned, the skeletal consistency and taxonomy could be useful indicators when deciding on adequate techniques for transplantation of marine sponges, especially again for restoration efforts in the future, because it's also very easily available information that can help during the decision-making process. The highest success was seen for species of Heteroscleromorpha, and same again in terms of restoration. If there's a chance, for example, to use species of this subclass, it may be recommended, because during the review they showed a higher robustness, and just the chance of success are probably higher than, for example, when using species of the subclass Keratosa. Also very promising, mounting sponges on cemented surfaces worked very, very well. Also what we've seen for Chondrosa reniformis, for example, in our own trials. I do want to mention that I excluded experiments on natural substrates in the review, but I think it's also a very promising technique that really needs more research. There was unfortunately only very few studies that investigated this technique, but the results seen in those trials were actually very promising. And I think, especially in terms of restoration, it would even pose lesser disturbance of the natural environment. And then, last but not least, the conclusion. I think now that we're in the UN decade of ecosystem restoration, There's the highest attention we ever had on habitat restoration, and probably also the highest available funding in the moment. So I think it's really important that we now try to identify species that actually enhance habitat restoration, and then intensify experimental trials. And I think this is also where my thesis comes into play, where it gives interesting and useful information, because in the end the approach should be to select and upscale those most promising techniques to have a successful restoration outcome. I do think it is very important also to mention that it's not only important to know the right technique, because I focus a lot on the techniques, but also to understand the ecology and the natural history of certain species, because you really need a holistic approach to have a successful outcome. Otherwise, you can choose the right technique and the right material, but you transplant the species to the complete wrong environment and your restoration will still fail. So it is really important to have this holistic approach to have a successful outcome in the end. All right. Thank you very much for listening. Yeah, I'm available for any questions.
Questions for you. I don't know how much it is known. You mentioned that there is a limitation on the knowledge of sponge species in general. What do you know about the life cycle and ecology of the species you targeted? And if you know if there is any analysis or how their life cycle or their growth compare to their growth in experimental settings or for commercial techniques?
Yeah, okay. In general, what I meant, the lack of knowledge is mostly that they're underrepresented in terms of example. There is information on many species, on the ecology, on the natural history of sponges. But for example, when we talk about global conservation status, there's only like a couple of studies that ever assessed sponges. It is, of course, a very, very challenging topic because the species are also very different from each other. And but yeah, just in general, what I meant is that with the restoration of certain key species, there's still this underrepresentation because we do know that sponges have very important ecological functions, and they, for example, increase structural complexity in certain habitats, giving habitat for certain stages of other species, for example, for fish larvae, etc. So this is a very, very important aspect. We have known this for a long time, but we're just starting now to implement it also in our restoration actions. The species, for example, that were cultivated here, it's very hard to put in a context because, as you saw, the growth measures I did, they were for a very short time span, but the experiments are actually going on. So it's going to be a long-term project. It's going to be very interesting, like you said, to compare it also to the natural populations, to have this comparison, which is very important, especially for cultivation, if you want to rear biomass or if you want to harvest, for example, secondary metabolites. Because then, of course, it is more important that you have a strong growth and to find the perfect environment. As I focused more on restoration, I think it's also important to say that I think in this term, it's maybe more important to look at the reproductive cycle and if sponges actually reproduce. For example, we went back to the spongia lamella transplants after 5 years, and we actually observed visually 2 of them. They looked very healthy. Now it would be very interesting in the next step to see if they actually reproduce, because for restoration, the growth may be a bit more neglected than, for example, the reproduction. You can have many smaller fragments that are able to reproduce, that of course enhance restoration more than if you have big experiments that don't really reproduce. I hope that kind of answered the question.
Yeah, it's enough. Thanks. What I'm interested also to hear a little bit more about the restoration process. How does it normally work, and which other elements are considered, and what is considered a successful restoration?
For now, there's like 2 very interesting projects, for example, in the Mediterranean Sea. One was the IMBRSea project, and there's another one coming up now that actually look into sponges as key species. And to have a successful restoration, it is of course very important to have a baseline, to know what is actually the status that you want to go back to then in the end assess if you actually achieve this goal. Like, you really have to understand the natural history, and also how the ecosystem was shaped before it was altered, for example, through bottom trawling or other impacts. And it is also, of course, very important that those stressors have been resolved. Like, you can try to restore something, but if there's still these pressures that depleted the original population, there won't be a good outcome. At the same time, as I mentioned earlier, it's also important to have like a very holistic approach, not only to choose the right method, not only to know the environmental factors that come into play, but you have to really put it all together to have a successful approach. And in terms of restoration, there's also one very interesting restoration project in the Florida Bay, where they're restoring sponges exactly with the technique I described, like sponges mounted on cemented bricks, and they actually put like little fragments, and they drop these bricks all across the lagoon, where they have been depleted very early. And it's very interesting because they have seen also increases in other crustaceans that were usually fished before, and they were completely gone. So there was this economical loss of certain fishes, and now it's actually repopulating itself because they started restoring the sponges, which I found very interesting.
Nice, very interesting. Thank you. And maybe just by curiosity, do you know if in this project did they also assess the eventual impact of these artificial structures once they are introduced in the environment? Like if there is an impact on the physical oceanography, on how the currents behave, so that might affect the nutrient flow and so on?
That is a very good question. I would have to look it up with these projects specifically. Like, I wasn't part of these exact projects. I know they exist. I think it would be very interesting. What is, of course, also always a delicate thing with restoration, also with cultivation. Most of it, especially in the past, was done using different kind of plastics. Even we or the university here has used this form of epoxy putty, because for the moment that is what works. But of course, it would also be very interesting to limit those effects of like especially alien materials. And as you said, it's for sure very interesting to also look in the change of currents and of the local regimes.
Okay, thanks. Do I have time for one very last question, Tim?
A very last one, then I don't have to ask that many anymore. Go ahead.
Yeah, it's another curiosity of mine. I actually don't recall if this was mentioned on your previous slide, but you mentioned the Asian use of sponges as bath sponges, and now the exploration of the modern exploration of metabolites. But do you have any idea if on the level of impact when sponges are harvested for aquarium?
What I found on this is really, it seems to be the lesser impact. Like, now at least a few sponges already made it into some protected lists. Like, for example, sponges are still missing on the IUCN red list, but there, for example, in the Barcelona Convention, certain species of sponges are mentioned already where the trade is regulated, but this is only for very few species. And there comes into play what I meant earlier. Like, we don't really know too much about the conservation of sponges. Because they're very widely distributed. They haven't been looked at for, or at least not as much as, for example, coral reefs. So they've really been a little bit, not neglected, but they just have received less attention. So we know also less about the conservation status. And then, of course, the trade is more open. And I think especially in like tropical regions, there's sponges, and I've seen online that there's sponges for aquariums. But I do believe it's the lesser impact, at least at the moment. It is also to say that the bass sponge exploitation has heavily reduced just because the population decreased. Like, the decrease during the 20th century was immense. Like, it really dropped nearly to zero. Like, even some countries halted in the 90s the sponge trade because the impact was too big and there was just nothing to harvest anymore. Because for bass sponges, it's also only a few species that can be harvested.
Okay, thank you very much. Congratulations again.
Thank you.
Okay, thank you, Luana. I also have a few questions. I wrote the whole list of questions, so I will select a few. I was missing something on genetic information, because if you just transplant and use them for restoration, don't you have the risk that your gene pool gets quite impacted? And what could be the potential risks to that, for example?
Very interesting question, and I think you have a really good point that you do have to be careful. Although I think it's also very challenging for a restoration project in terms of the analysis and the process to look into these aspects. And for sure, if there's a limited diversity in the genetic pool, there could be problems, for example, with disease outbreaks, which we have seen in the past in the Mediterranean, where populations, for example, have been heavily decreased due to exploitation, and then disease outbreaks really impacted the populations even more than it did before. So I do believe we have to be really careful, and it should be put in consideration, but I also think it's a very challenging aspect for the project.
Yeah. Although, yeah, you could say, okay, we take by default at least samples where we do barcoding, just to make sure that that we have a track record of what we actually use in certain restoration projects, and also to see, for example, what is the plasticity of the. So that's one comment I had. Kind of another one is maybe a question: what is the gross economic value of the let's say of the sector, the economic activities related to sponges? Could you?
I would have to look it up again. I can't give you the concrete numbers.
The question a bit related to that is, okay, is because these are experiments that are quite in an early stage. The question related to that, if there's indeed a high economic value, then you would say, okay, well, people wait for the outcomes of these kind of experiments and not just go ahead and just do whatever they want, as long as they can harvest sponges, because there's no regulation at all. So don't you think that it's a bit slow, the connected to the potential economic value of
it is the tricky thing. It has been tested for many years growing sponges, and there is a certain economic value. Like I mentioned, the trade really dropped, especially during the 20th century, but the demand, people have estimated, is still there. And there is actually 2 commercial sponge farms, maybe a few more. One is in Zanzibar, the other one is in Micronesia, where they do harvest sponges and sell them. For example, one bath sponge, a natural bath sponge, is sold for approximately $40, which is actually quite some money.
They don't grow them, eh?
They do grow them.
They do grow them. It's not just harvesting from the natural environment.
No, they grow them on rope systems. The problem is with this approach, it needs certain maintenance, and it's if you would do it in the Mediterranean, I think the cost would just
Yeah.
be too high. So many people still investigate these processes, but I think we have, at least in certain cases, we have found the techniques. It would also be very interesting to find out how to maintain these systems and to make it more manageable.
Yeah. Well, I had more questions, but just maybe a small final one. What about you talk about growth? Yeah, and you showed there one graph, where it's but how do you measure growth in this case? Because we deal here with sponges with complex 3D structures, where growth is not just a linear thing. It's not like a fish which becomes larger, or the weight, for example. So how to deal with growth when you talk about complex 3D structures, which are important, for example, for associated fauna then as well.
So what we did, we used Agisoft Metashape, which is like a 3D modeling program. So we could actually create a model of each sponge. And then what we did is the surface area that we measured. And like I said, also the volume. Because like I said, if you only look at one, you can lose certain aspects, because you mentioned it's like a 3D shape. So even if one of them increases, doesn't mean, for example, the volume or the structure complexity increases. This, of course, gets more challenging if you, Chondrosia was relatively easy and simple, because they have this like oval kidney shape normally. But for example, with Axonella polypoidis, which is a branching species, it gets more challenging and needs a bit more work on the modeling perspective, but you can actually excuse me, create like a very nice model of the sponge that you can then analyze. Also highlight the growth, for example, in like a video where it grows just on like a certain tip, for example, you can highlight where
The hard thing is to compare two different species. That's what you say. You have a typical kidney-like shaped thing, and then a complex branch. So how to make comparisons, but between them, for example, whether on success of growth, for example. So it's just from interest, let's say, and what could be done. Okay, good. Well, I don't see any questions on the chat. I don't know if the other presenters have questions for Jan. There's time for a very short one. If not, nobody just puts on his camera or microphone, then I think we can conclude this first presentation here. So thank you very much and discussion, and we'll be back in a few more minutes for the next presentation. Okay, good morning again. Second presentation of this session already is a presentation by Juliette Vilshanu on, and she was assessing the effectiveness of different transplantation methods of red Corallium rubrum. So very connected to the last presentation about sponges, but now we switch to the red soft corals. So Juliette, the floor is yours for your presentation.
Corallium to help future restoration projects. So I did that by first compiling information on transplantation techniques and larvae enhancement experiments of Corallium, and then I performed a case study of 6 transplantation techniques of Corallium in the Ligurian Sea. I analyzed their effectiveness, and I also analyzed if transferred shallow colonies and juveniles could survive in deeper waters. So I will explain to you how the fishing island here. 2 of those transplantation experiments were done under crevices with the transplants facing upside down. And the component that was used to glue all the transplants on the substrate was a 2-component epoxy putty. That takes a few hours to dry. So the transplant maintained in place while it dried, we put a polystyrene packaging here that was removed after a few hours for one method. In another method, we put a PVC grid that was fixated on the substrate on top of the epoxy putty with metal pinches, and the transplants were put on top of that. 2 other transplantation experiments were done with transplants in an erected position on rocks. So one just with putty, no extra material, and the other one also used the PVC grid fixated on the substrate with metal pinches. The second row of experiment NPA, and it was a transfer from shallow waters to from 30 meters depth to 7 centimeters depth to see if shallow colonies could survive in deep environments. So the shallow colonies were transplanted with epoxy putty on an open wall, as you can see on picture E. And then we also transferred shallow larvae that were collected on 6 PVC tiles that were put on the roof of a cave in spring 2016 that stayed there for 2 years to collect 2 cohorts of larvae and then move very carefully at 7 meters depth and put on a vertical wall next to red coral colonies. The data collection was done via underwater and photographing samples. Then I created a sequence for each pictures of transplants and PVC tiles to identify the time, and then to assess the effectiveness of each colony's transportation, analyze the survival and loss rates of transplants, after analyze the appearance of new branches, so the pattern of growth variations, the height was measured for each transplant with IMBRSea software. And for last, to analyze the skeleton structure and the health of the tissues, we collected apical fragments of the transplant to analyze them under the scan electron microscope. Regarding the larval enhancement experiment, the number of shallow and deep settlers were identified on each picture. And after, the survival rate of shallow settlers was analyzed and the density of shallow and deep settlers as well. First, I will talk about the review I did on past transportation larva enhancement experiments of Corallium rubrum. So I found that the first transplantation for this species was done in 1979, but very poor results, 0% of survival, unfortunately. But in 1992, the first large-scale experiment was done with high survival rates, and it used resin for the first time to glue the transplants on the substrate. After this experiment, resin was successfully used on Corallium substrate until 2000, and since 2000, only 2 experiments were made, also using epoxy putty. So from the 10 publications that I found, all the transportation were done with the colonies in an erected position, and only one did an upside down position, but it was in an artificial substrate. The survival rate after 2 or 3 years was of 71.5%. Regarding the larva, we say that it's a recurrent method to study settlement and recruitment density of Corallium rubrum since 2000. And there is an average settlement density of 4.71 settlers per decimeter squares. It depends a lot of the position of the tiles, and the best reproduction results were shown on the caves and roots. But over a long time period, this high density drastically drops to 0.9 colonies per decimeter squares after 21 years. Now I'll talk about the results of the transplanting techniques in shallow waters at Galinara Island. So the hypothesis was that the survival rates will be lower under detachment, higher in the crevices. And that's what we observed, because the higher survival rate was for the grid on rocks method with 60% of survival, and the methods and the crevices, the survival rate was quite similar after 4 years with 30% of survival. The difference is that for the polystyrene packaging method, the loss was very quick in the first weeks and months compared to the grid and the crevices method, whereas the loss was very sudden and a big loss, but after 1 year and a half only. On figure 7, we can see the detachment of the transplants for each method through time. And what we can see is that most of the loss of transplants can be explained by detachment, by method failure, and not by 50% of all transplants. Without surprise, the methods that had the higher detachment rates are the ones that were done on the crevices, and that can be explained as before, with the polystyrene packaging that was not effective enough to hold the glue. So we can see here that the fragment detached itself just 1 day after the transplantation was done. And the problem with the grid is the risk of detachment of the whole grid. Then it's a sudden loss of all the transplants attached to the and the crevices. The grid also detached itself on the grid on rocks method. So when I analyzed the appearance of new branches, we were hoping to see a positive number, so branches growing, but what we saw was the opposite, because in every method, most of the transplants lost branches, so the branching difference was negative. And this is confirmed by the results of the Kruskal-Wallis test that showed no significance between the different transplantation methods and difference, showing that the branching difference did not matter which method was used. And all methods confounded, 56% of transplants lost branches with an average of 35.8% of loss, and only 8% gained branches. We can see this loss of branches on those two transplants, for example. So here it is just after transplantation, and that is not even 1 year after. We can see for these transplants that he lost all its branches, unfortunately. And the other one lost so many branches, it became so small that after 3 years, it was affected by epibiosis completely. When I measured the height growth for transplants, we're also hoping to see an increase in height. But unfortunately, it's the same as the branching. We saw a decrease in height for most of the transplants. We have to take into account that the height growth could only be measured for the transplants using the grid method, because the grid was used as a reference bar to measure the transplant. So 80% of the transplants on the crevices decreased in size after 1 year of transplantation, and 20% continued to decrease after that. Regarding the grid on rocks method, only one transplant decreased in size significantly after 1 year. I did a correlation test to see if there was a correlation between the branching difference and the height growth, and the test showed very poor and non-significant correlation although. The strange is that red coral is a hard sponge. It doesn't move that much normally, and we saw a branch decrease and we saw a height decrease. So it made us think what happened skeleton, and if we could see any form of indication of might have had apical fragments of the transplant in Galinara Islands and with mental pinches. So here we can see the clean cuts done by mental pinches, and being seen under the SEM microscope, there's no unusual pattern to be seen. But as we look into other apical fragments, so G, E, and F, it's from a transplant, and when you look at the apical part, you see a smooth pattern that can suggest an alteration occurring, because normally the pattern is supposed to be like a skeleton. So this is a 60-year-old fragment as a reference. Another apical fragment showed that two unusual structures, where we can think that it was a branch growing before. There is a protuberance growing, and when you look at closer look, it looks very much like the 60-year-old fragment, which could make us think that there's a regeneration process going on where a branch was before attached. To compare the transplants, the fragments of transplants, we also collected fragments from natural colonies in Portofino NPA at 25 meters depth. And when we collected them, some of them were already getting loose by their own from the main colony, and we didn't have to use mental pinches to count them. This is one of those fragments, and we can see at the bottom part that was attached to the main colony a very particular pattern, same thing, protuberance, but this time very smooth. We can think that there is a detaching the cell from the colony. So with this Clarexis analysis, we can hypothesize there is a fragmentation process going on in Corallium of shallow populations. All the structures were not similar to what could be in the future. Now moving to the transfer of shallows, it was very surprising because it was quite successful. After 1 year and a half, there was 82% of survival from the shallow colonies on the open wall, and all the loss was due to detachment, so to method failure. On the opposite of Galinara Island, the transplants were looking healthy and there was no branching decrease, only one transplant lost branches. Regarding the settlers, even though it was a very problematic way to move them down with the tiles, 50% of them survived on tile B. Only 2 tiles could have been identified in the service, and 32% of them survived on tile A. When analyzing the settlement density of settlers, it was found that deep settlers settled on both tiles in the cohort of 2018 and also a cohort of 2019. You can see it's the red dots and black dots on the pictures here. The total average settlement density of all settlers confounded was 1.98 settlers per decimeter squares. So what can you say about this study? First of all, the study enters into the United Nations Restoration Decade that was announced in 2021 by apporting information to restore the engineering species of Corallium rubrum. The geomorphology of the cliffs is a very important variable to be considered when the restoration plans are designed for the species, because as you can see, the inclination of substrate can strongly affect the survival of transplants, but also of the parent. The technique here has to be in the crevices method to reach a higher percentage of survivors and guarantee an effective restoration. And list, here we report the first chapter in situ. It was reported in the laboratory by Russo in 1999, but since then, no other observations were done. It can be suggested that it's a net behavior from Corallium rubrum to cope with new environmental conditions and respond to stretch in shallow areas. Thank you very much for listening, and I'm here for any questions.
Thank you very much, Juliette, for the nice presentation. Let's move to some questions. I don't know, Luana, you have some questions?
Yeah, I feel. Thanks. Juliette, congratulations. Very nice presentation, very clear, like a lot your slides and the outline. Similar to Jan, congratulations, because I think you produced a very important document for risk assessment, management of the red coral, and of course for the establishment of the restoration plans. I have a few questions. I think you gave a broad outline on the main topics of discussion, but could you maybe elaborate a bit more on, based on your findings of the tested methods and the results that you got, which methods you think have more chances to be improved? The impression that I got is that there was a decline in survival rate on all of them, but you in your discussion you say that there is room for improvement of the techniques. What is your idea based on your results on that? Is there any method from the 6 that you think is more promising than others?
Yeah, so in my opinion, the grid and the crevices method, so when we transplant the Corallium rubrum, it's in natural habitat, which is in an upside down position, and the grid to really fix it, it's a very promising method. We just have to, grid is well fixed, so it fall, but it allows that, because results show that the best reproduction input and larvae settlement is when the corallium is in an upside down position. So if we transplant in an upside down position and improve the grid's fixation on the substrate, then there will also be better recruitment of larvae, because it has been shown that the larvae also goes up like this, and that's why the settlement is also higher when the tiles are put under roofs and caves. So I think that the grid and the crevices method is very promising. You just have to find a better fixation method on the substrate.
Okay, thanks. I don't know if I'm interpreting things wrong, but what was different between the testing of the methods and when you did the transplantation to that deep waters, and that was successful? So what do you think are the main differences there?
So I think that in deep waters, the red coral is less affected by extreme environmental conditions, because it has been shown that in recent years, the shallow populations have to face extreme temperature changes, temperature anomalies, and when you go in deep water, there is less current in this place where they were, there is less temperature anomalies, a lot of changes, and they're more protected in a way. The only thing that they could affect their survival, but it didn't in this case, is has sediment cover. There's more sediment in the deep, but in this case, it didn't affect them.
Okay, thanks. It's nice that you mentioned a reference about the possible coping mechanisms of adaptation that these species have that would justify such successful rate. But you also showed earlier that the species naturally can be distributed and found at different depths. I was curious when I saw the high successful rate to know if there is any information on the literature about the evolution of the species. So if there is any genetic study that already identify if there are differences in the lineage of populations who have been established many years ago in the depth compared to the populations that inhabited more shallow waters, or if this is something that still needs to be investigated?
The problem with this species is that it has been adapted for so many years, for millennia, that what we can compare it to nowadays, we can't say that it's its original state. We will never know how red coral was originally and is originally in deep waters and shallow waters, because we modified so much its population structure and how it is distributed, that what we can see nowadays is just a consequence of the harvest it has been affected by. So there is studies on genetics between deep and shallow, especially in the last year, since we found that there's some difference with the shallow populations and deep populations. But if that is a consequence of recent margin environmental conditions, or if it has always been like this because then they got separated by distance, it's hard to know. There's also a lot of studies on the dispersal of larvae to know how those can be, because they are more patchy distributed compared to shallow populations that are very dense. So there's a lot of studies also beginning in the last years to understand how these populations reproduce itself and manage to create more colonies, because it's a more distributed population pattern, and it's hard to understand how the larvae can travel in such a big distance. When we found out that the larvae actually moves upwards in the caves and on the roofs, it's under study.
Yeah. Okay. Yeah. I imagine there's a lot. Yeah, a lot to investigate on the behavior of the larvae and so on. Thank you very much. Congratulations again.
Thank you.
Okay, thank you very much, Luana. I think I have time for one or two small questions, maybe. I'm a bit surprised also with the former presentation also by Jan, yeah, that we're trying to avoid plastics and artificial stuff in the environment, and then what you see when it comes to restoration, I find it a bit dubious that we look at epoxy, we look at plastics, we look at a whole kind of things, a range of things, which actually many of them are kind of new, of which we even don't know what are the long-term effects of putting these in marine environments. So I'm a bit sometimes puzzled on the types of substrate that I used for a specific goal. So while at the same time, actually, you're also polluting kind of your environment again. So what is the cost benefit actually when it comes to these elements? Could you comment on that?
I completely agree with you first. And it was a discussion we had with my promoter and my supervisor that it is hard to find nowadays material that is not made of plastic to do these restoration projects. And I know that my promoter is actually trying to build an epoxy putty that's less harm for the environment, but it's an ongoing project and process. And I know they also think of trying to use other material than a PVC grade, but nowadays it's unfortunately it was a first tryout, it turned out to be good, but I think that's also one aspect that can be further improved. When I say the techniques can be improved, maybe trying to find another material than plastic, unfortunately. Maybe some materials then can degrade themselves after a few years once we make sure that the corals are Yeah.
Okay, the last part of the sound was a bit, but doesn't matter. I have another question maybe related to that. Is that do you think that the material actually does make a difference? Because what we see from literature, for example, when it comes to biofouling and other things, that it's actually the material does not matter. It's the structural. Okay, what is the is it a flat surface? Is it not a flat surface? And related to that, okay, what are the connected biofilms? And that has a much bigger importance when it comes to colonization than whether you use plastic or metal or glass or so that's maybe a second question, whether there was a look into, let's say, the surfaces also at the micro level. Okay, why does a certain species or a certain specimen attach better or not to certain surfaces?
I didn't understand the last part. Why?
Yeah. So my question is a bit giving that introduction on the whole biofouling thing. Yeah. Did you look into, okay, the call it the micro surfaces of these materials and associated biofilms to see whether this has an impact or not on your success of colonization or of growth? Yeah.
I didn't look into that, but what I can say is that in past transplantations, they also use porphyry bars to put the red coral in and fixate it with, and the porphyry bar also worked very good. So I think you have a point here that it's not the material more of the problem, it's how the structure is. Better, another material that's not plastic and not as pollutant than the PVC grade, but for sure it will be better.
Yeah, yeah, but there has not been looking into biofilms,
No,
for example.
No, I didn't.
Yeah, because sometimes I find these colonization or recolonization things. Okay, we should get back to a certain thing, but you also have to take your total assemblage into account, from micro to also associated faunae. What is the cause and what is the origin of certain things? Yeah. Well, I think from a time per point of view, we could discuss more. I have more questions actually, but we have to restrict it to here. So thank you very much for the second presentation. We will be offline for just 1 or 2 minutes, and then we go on to the next presentation already. Thank you very much.
Thank you. Bye.
Okay. Okay, good morning again. Third presentation already. We move from recolonization experiments and to complete different area that shows immediately also the diversity of this master program. And we move to a presentation by Laurie Chapuis on the interaction between Agarophyton vermiculophyllum and two types of bacteria. So, Laurie, the floor is yours for the presentation.
Thank you. So, hello, everybody. I will present my master thesis about the interaction between Agarophyton vermiculophyllum and two bacteria, G-Nord11 and G-Man6. So, first, Agarophyton vermiculophyllum is come from the Pacific Northwest, but it's an invasive species. So here you see the native species, and in red the non-native. It invests a lot of different area, and it takes with him some bacteria we can find in the epimicrobiome, and in this bacteria you can find G-Man6, which is a pathogenic bacteria, and Gimandoalteromonas, precisely it's Pseudoalteromonas arctica, and it's a non-pigmented because you have two groups in the Pseudoalteromonas, and the protector, it's a pigmented one. So you have the picture. Then G-Nord11, we don't know exactly which Pseudoalteromonas it is. So in my master thesis, I try to answer many hypotheses. So if we look if the bacteria are pathogenic, so at the beginning I had two of them for vermiculophyllum. If the other bacteria are protective or not, if it's the effect of the pathogenic and the protective effect come from an excretion of effective metabolics, and if the protective bacteria inhibit or kill the bacteria, and finally if the quorum sensing plays a role in the interaction between pathogenic and protective bacteria. So for that, I had many experiments. Some are bioassay. So to prepare this bioassay, I add 2 milliliters of water in whale plates with the tips of Agarophyton vermiculophyllum, and I add antibiotics to try to eliminate the maximum of the bacteria on the wait 3 days, and after I rinse 3 times, I watch them on the binocular, and I add the bacteria and all the molecules for quorum sensing. I wait 7 days, and I relook if the bacteria are if the tips are bleaching or not, like this image. See the white tips here and the brown here, and I count how many white and how many brown I have. So it gives me these results. All these are potentially protective, and Cordia algicida and the other pathogenic bacteria. And we can see that not all the protective bacteria are really protective with Cordia algicida, only 2. But with Pseudoalteromonas arctica, all were protective. For the following experiment, we choose to use only G-Nord11. At the beginning, we use GNY6 and J1I, I1, but we didn't see some results. Another bioassay was to know if we have a limit of in the dissolution to have an effect on Agarophyton. And with G-Nord11, we have the dilution 3 and 6, which were different. but the effect is not really clear. We don't have a really clear tendency. But with Arctica, we have a really tendency, but nothing was significant. So, and the dilution 6 was the limit for the have an effect of G011, and for Arctica it was the dilution 4. Arctica is G-6. And another bioassay was to look if the bacteria combine, if we combine the bacteria, if we have a dose dependency on the effect. So all the green is the tips where not bleaching. If we compare the control with A, it's for Arctica, and P for G011. And if we combine, if we look the control with Arctica for the different dilution, we see it has an effect. So the control don't bleach. And if we combine G011, the pure culture, with Arctica with the pure culture, they don't bleach too. We see here with the control, it's 1. It's the p-value of the experiment. But if we look the G011 with at the dilution 3, so a smallest dilution, we have a bleaching. So with Arctica, they don't have difference, but with the control, they bleach. And it's the same for all the other, the dilution 4 with pseudoalteomonas sp, but in combined with Arctica with the different dilution too. And the blue, it's the tips where to bleach to be different. They have a little bleach, but not enough to be different with control, but 2 to be different with Arctica alone. And to do that, we have to calculate the quantity of cells, to combine the quantity of cell with the absorbance. So we count the absorbance for we calculate the absorbance for different dilution, and we look on the microscope. It gives that beautiful picture, and each picture were count. The cells were counted by IMBRSea, and I calculate the quantity of cell per dilution, and gives that. And we know that G-6 is 10 times higher than G011. The absorbance of G-6 is 60 times higher than G011 for the same quantity of cells. And we look the emissions of molecules. So we put a biofilm on agar, and we do some hole where we put the opposite bacteria. So here on the biofilms, it's Arctica, and in the hole it's G011. So you can see the visible effect around it, inhib or kill, we don't know yet, the Arctica. So after, to know if we have an inhibition or a killing bacteria, we put the bacteria on transwhale plate, and so that's the transwhale. You can observe here the filter. So at the bottom, it's a normal plate where I add approximately 100 cells per whale. I filled up until the top, and on the top I add the quantity of cells. So first pure culture, and until to have approximately 100 cells. And I did the absorbance and T3, and we looked the dead and live cells at T3. So for the cell density, we don't see any significant difference with the different dilution. But for G-node 11, we can see the control grow a little more than the other dilution. It's possible that because we have more nutrient available, since the control is just liquid medium. For the dead life treatment, so the live cells, it's in green, and the dead, it's in red. You can see clearly a difference on the microscope. And so here too, we don't have any significant difference between the G-6 and the G-node 11 for live and dead cells. And we did the same with the exudate, and any difference was apparent too. For many experiments, we did the same with exudate, but nothing was gives us some good results. So to conclude that, so I have a pathogenic bacteria for both, so Cordia algicida and Pseudoalteomonas arctica. Not all the protective bacteria are really protective with both pathogenic, but they are all protective with Pseudomonas arctica. And we don't know if the pathogenic effects come from the excretion of the effective metabolite. And it's possible for the protective effect come from an excretion of effective metabolite, but it's not sure since the exudate didn't give us the clear result. Apparently, the protective effect is more an inhibition than a killing, and the quorum sensing play maybe a role in the interaction, but we tried to use 2 molecules, but it didn't work because at the beginning we saw that G-node 11 was Pseudoalteomonas piscicida, and it was not the case. And the 2 molecules we used was for Pseudoalteomonas piscicida. And finally, we saw we have some problem with reproducibility, which can be explained by the fact that the alga fresh alga was collected for each new experiment, and the water become cooler and cooler, so it can affect the alga and the bacteria community. So thank you for your attention, and if you have any question, try to answer it.
Thank you very much, Lori, for the presentation on a more abstract subject, I would dare to say, because yeah, it's less easy to visualize previous presentations. Just checking, Luana, do you have any questions for Lori?
Yeah, a couple. Thanks, Tim. Lori, congratulations.
Thank you.
I find it a very interesting topic. Investigated and generated valuable information on interaction of bacteria. A lot yet to be found out. Maybe well, I would like you to maybe share what would you if you would have the chance to continue with that line of research, I would like to know what would be your following approach.
So,
and maybe briefly explain for those who are not acquainted with bacteria interaction, what is the quorum sensing?
So if I can continue, we can search which molecule is used by G-Node 11 to reduce the possibility of Arctica to infect the alga. It can be protease or antibiotics. We don't know in which direction we can go. And quorum sensing, it's a system where bacteria have a communication between them, and they associate together to excrete molecules. It's a system do with it's genetic. So they really, when they have enough bacteria, they say, okay, we can do something together genetically.
And do you have any idea on possible influence of other microorganisms or even small invertebrates in the communication of bacteria? If they would play also a role into this kind of interactions or the susceptibility of infection by microalgae? Or in other words, would there be any other microorganisms that could play a role in the protectiveness or susceptibility of the algae to the pathogenic bacteria?
So for Agarophyton vermiculiphyllum, at the beginning, the algae had a lot of epiphytes, so it can interfere with the communication between bacteria and bacteria with the alga. And yes, the epibiome, a lot of different bacteria are, or protists or diatoms are present around the bacteria, so they can play a role. We know if we put antibiotics that don't kill all the different organisms, on the bacteria, on the alga.
Okay, so, I'm just wondering if there would be an alternative to the use of antibiotics, if there could be any organism that would live associated with microalgae, for example, that could also produce itself metabolites or even carry bacteria that could be protective against the pathogenic bacteria. So I don't know if that is a line that are being investigated already.
So for this alga, they show that it can attract the protective bacteria. So maybe with other microorganisms or like that.
Okay, thanks. That was interesting. I rest my case, but congratulations.
Thank you
for your research.
Yeah, thank you very much, Luana. Well, I actually would like to switch from the bacteria more to the algae, because you didn't say a lot. You briefly introduced, but when I search up around here, the algae, it's a species that is in the aquarium world quite widely used and also sold. And actually, I would like to elaborate a little bit on that, because can you say also that if it's used in aquarium settings, for example, that these bacteria also play a role there, for example, whether there's any information on the dispersion, for example, of these bacteria also through, and whether there is literature on that as well?
So for G-Node 11, we don't know, since it's a non-descripted bacteria. Yeah. So it's difficult to know if it's well distributed or not. But for G-6, apparently we can find it quite easily, almost everywhere.
Yeah, so also in Aquaria, you're
In Aquaria.
Yeah, because that's one of the things. Because a bit related to that would be my question. Okay, and how far? As I know these metabolites, for example, and these molecules can potentially be interesting for direct applications. But could you, for example, also make now one step further in your master thesis, your research? What is the next step? What is because this is a master about biological resources. So we're also looking into, okay, what can they be used for? To use this word. Eh?
If we can know which molecule it's excreted by G-node 11, if it's, for example, an antibiotic, we can maybe use for human for later.
Yeah. But most likely it's an antibiotic. Eh? Because if you see a direct, then
antibiotics or maybe protease.
Yeah. Yeah. And did you look into other, for example, did you look on the effect of other, because bacteria are not living, just to make sure there's only one, it's very hard. Eh? Did you look at the impact also on potential other bacteria?
So for the experiment on the agar, I tried the other bacteria, but we didn't see any effect or clearly effect as a G-node 11. It's why we choose to work only with G-node 11 and Pseudoacteomonas.
Yeah, and so because from a direct then in the research you're doing in that research group, is it the aim that they do this test for all kinds of is it methodology, or is it with a clear goal to go into one direction?
So for G-node 11, it's to understand better which molecule it's excreted.
Yeah.
And maybe for other bacteria to search a little more and on them to know. We don't see on agar if they excreted something or not, but maybe they do it. So many direction. We can take many direction.
Yeah. Okay. Good. Voilà. I think that's, for me, that's enough. I'm just checking the chat as well, but I also don't see any questions there. So I think we can conclude here and nicely stick also to the schedule. So thank you very much. And we'll be offline for a few minutes, and then we'll back with the last presentation of this morning.
Thank you.
Okay, good morning, and welcome to the last presentation of this exceptional meeting of the IMBRSea Master Program. So we have as the last presentation Olivia Saiz Munoz, which will talk about macrofauna communities in a coral reef in the Isle of Gorgona. So you have now the floor, Olivia.
Okay, thank you very much. Well, good morning all. My name is Olivia Said and I'm going to present the thesis project that I've done with La Universidad del Valle in Cali, Colombia, under the topic Size Structure of the Macrofauna Community in La Azufrada Coral Reef. The presentation will follow the same structure as the manuscript, so I will address these 6 sections in the following order. Corals are considered foundation species because they host a wide amount of organisms. Most of them are comprised by macrofauna. Macrofauna also entails the highest abundance in these formations, and they also have key roles in food webs. However, despite their importance, research is still very scarce, and this is usually linked to the small size and creative behavior of these organisms that usually hampers the sampling methodologies. To characterize macrofauna community, it's important to evaluate both habitat and body size. On the one hand, habitat has been considered a key factor in structuring the composition of benthic communities, and in this case is particularly important because coral degradation is a worldwide and growing phenomenon that is characterized by a habitat transformation pattern in which there is a switch in the dominance of the type of coralline substrate from live coralline substrate to dead coral fragments that are usually colonized by amphibians. On the other side, body size is also a very important attribute because it affects ecological processes such as metabolic rates, generation times, and predator-prey interactions, and therefore changes in the community size spectrum might lead to the alteration of the direction and efficiency of energy and organic matter flow through the food webs. However, mostly of this information remains largely unknown, still again due to the scarce research. In the extratropical Pacific, studies were conducted during the 70s, mostly in Panama and Colombia, and most of them were focused on the description of species belonging to a Capoda order that are organisms intimately associated with the branching coral species of Pothylopora. Particularly in the Colombian Pacific, most of the studies had been conducted during the 80s and Isla Gorgona. Therefore, the aim of this research was to characterize the macrofauna community associated with 2 different coralline substrates in a Colombian Pacific coral reefs. So, 3 community attributes were evaluated: biomass, measured as wet weight, density, and community structure, considering high taxonomic levels. So, 2 objectives were addressed. On the one hand, how these attributes changes in between live and inner coralline substrate, and how these attributes vary within 5 size group in the 2 coralline substrates. Okay, the sampling was conducted in Isla Gorgona. In this island, different coral formations are found. One of them is La Azufrada. La Azufrada coral reef is indeed one of the most developed coral reefs in the Colombian Pacific. And in order to characterize the macrofauna associated to this coral reef, 40 samples were collected in 20 points randomly distributed in the coral reef. Therefore, 2 samples were collected at each sampling point, one corresponding to live coralline substrate defined as entire Pothylopora colonies, and other corresponding to inner coralline substrate defined as coral skeleton devoid of live coral tissue. After the collection, these samples were placed in a jar containing seawater and defaunated. So after the defaunated samples, these samples were filtered through a sieving shower containing 4 sieves arranged in a certain order. Therefore, according to the size, macrofauna were retained and sorted into 4 size groups: 1, 2, 2, 4, 4, 8, and larger than 8 mm group. After that, wet biomass was estimated for its size group, and macroinvertebrates were counted and classified to higher taxonomic levels and preserved in 96% ethanol. As organisms belonging to a 0.51 mm group were very small, a slightly different processing was conducted for this size group. Therefore, after filtering the samples to the sieving tower, the remaining solution was poured again into the Nalgene filtration system, which contained 2 meshes, one of 0.5 and the other of 0.2 millimeters. It's important to mention that in order to be able to do comparison, all data were standardized to 100 square centimeters, Biomass and densities follow the same, their analysis follow the same structure. So firstly, differences were tested between coralline substrates using a PERT test or a Wilkinson test if the parametric assumption were not met, and then differences were tested between size group using an ANOVA of a linear mixed effect model, and this was followed by a two-case post-host test in order to evaluate pairwise difference among size group. For the community structure, densities per taxonomic group were used. So in order to test differences in community composition between coralline substrate, the anosine test was run, and this was followed by the simple analysis to evaluate how its taxon contributes to this dissimilarity. Our results revealed that wet macroinvertebrate wet biomass was significantly different between coralline substrates, being remarkably higher in life than in coralline substrate. Indeed, mean biomass in live coralline substrate was more than 12 times greater. Differences were also found between a size group in both coralline substrates. Indeed, all pairwise comparisons were statistically significant except the one marked with a red asterisk. And it should be mentioned that in both coralline substrates, biomass increased as the size of the organism increased, and the contribution to total biomass was made by the largest size group, which was more noticeable in live than in inner coralline substrate. Regarding density, however, differences were not significant difference were not found between coralline substrates. Still, it should be mentioned that some samples belonging exclusively to the inner coralline substrate had really high densities. When densities were studied among size group, differences were also observed in both coralline substrates. However, the behavior of this community attribute differed between coralline substrates. On the one hand, in live coralline substrate, contribution to total density were more or less balanced among size group, and the highest density were comprised by the intermediate group, the 1,2 and 2,4 millimeters. However, in inner coralline substrate, the highest density, the density follows a monotonical decreasing pattern because it decreases as the size of the organism increases, and density contributors were not homogeneously distributed, but mainly contained in the 0.51 millimeter groups. With regard to the community structure, it's important to mention that in both substrates there is a dominance of a single taxon, and that macrofauna community, the composition of macrofauna communities were statistically different between coralline substrate as revealed by the anosine test. Indeed, this difference accounts for 78% between substrates. Caridia, the highest contribution to this dissimilarity was done by Caridia because although it is present in both coralline substrates, it has a very different relative densities. And the second highest contribution to this dissimilarity was done by Tanitharia. In live coralline substrate, it's important to mention that the dominance of a single taxon is also observed in all size groups, except the 0.51 millimeter group, and it's especially conspicuous in the intermediates group, where Caridia densities exceed 70%. However, in inner coralline substrate, again, this dominance of a single taxon is observed in all size group, and it's especially remarkable in the largest size group, where the dominance of Phylloidea accounts for more than 80%. During the discussion, our results were compared with other studies. However, it is important to mention that the different methodologies used during the sampling and the processing hampers these comparisons. In our case, biomass showed the most conspicuous difference between coralline substrates, and these results have been also observed in other studies where live coralline substrate have higher densities, very high biomass than inner coralline substrate. However, contrary results have been also found in the bibliography, such as, for example, results reported by Kramer et al. That said that biomass were very similar between live and inner. When biomass was evaluated among size group, in live coralline substrate, our results agree with the ones reported by Fraser et al. And it's important to highlight this coralline substrate because In this case, biomass is not only compressed in the largest size group, but also in a very specific taxa, in organisms belonging to the Trapezia and Alpheus genera. Therefore, total biomass in live coralline substrate is comprised by the capods that are obligate symbionts of the Pothylopora colonies. It should be mentioned that results obtained in this study regarding inner coralline substrate and the one reported by Fraser et al. Differs because Fraser et al. Report that biomass was not dependent on size in inner coralline substrate. Regarding to density, also similarities and differences were found in the bibliography. It is important to mention that these inner coralline substrates yielding very high density values in inner coralline substrate are usually due to the size of the mesh used for retaining the small size class, because there are some taxa that are particularly abundant in size groups below the 0.5 centimeters, such as, for example, the Capoda. When density was evaluated among size group, it was observed that our results agree with the ones reported by Fraser et al., and special attention should be paid to the smallest group in inner coralline substrate, because hyperabundant samples occur exclusive in this size group. Therefore, in some inner coralline substrate favor the presence of high abundance of small invertebrates lower than 1 millimeter. With regards to community structure, again, this high difference regarding the community composition between both coralline substrates is consistent with literature, because, as mentioned before, habitat is a key factor structuring the benthic composition. It should be mentioned that in our case, these differences are due to taxa that are present in both substrates, but are strongly correlated to only one of them. Also, it is important to mention that Galinara of crustaceans have been observed in both coralline substrates, and the importance of crustaceans has been stressed by several authors in macrofauna communities associated with coral formation. In live coralline substrate, it's important to mention that all the size groups were dominated by crustaceans. However, different orders were found according to the size group. Therefore, the smallest size group were dominated by Pericarida, and the remaining size group were dominated by the Capoda order. This is important because most of these organisms are facultative, are obligate symbionts, and therefore rely heavily on live coral tissue. In inner coralline substrate, however, this abundance of crustaceans are also observed in the smallest size group, and it is conspicuous because in the crustaceans dominate the overall assemblage in inner coralline substrate, but they only dominate the 0.51 millimeter group in inner coralline substrate. Here, it should be mentioned the dominance of Philidea, because they have identified in other macrofauna studies as disturbance, as they are usually linked to lower diversity index. Therefore, to conclude, when studies of macrofauna communities associated to coral formation are done, it is important to consider, on the one hand, both substrate, live and inner coralline substrates, and on the other hand, both perspectives, the overall assemblage and the distribution within size group. Specifically, transitions from live to inner coralline substrate in Lazufrada coral reef might lead to lower organic matter contribution, a higher amount of smaller macroinvertebrates, and an increase in the macroinvertebrate diversity. This research, it's important to mention the relevance of this research because it's one of the few analyses conducted in the Colombian Pacific where these three community attributes have been considered, and most important is the first analysis not in the Colombian Pacific but also in the tropical Eastern Pacific regarding the distribution of these three community attributes among size group in live and inner coralline substrates. Thank you very much for your attention.
Thank you very much for the presentation. Yeah, time for questions, I would say. We still have a few minutes left in this long session. So I think Luana has, again, some questions. At least that's what we hope, of course. So Luana, the floor is yours.
Thank you, team. Congratulations, Olivia, for your presentation. I don't know, maybe you can stop sharing your screen. So
Yeah, sorry.
no problem. Otherwise, I was talking to myself.
Yeah, I completely forgot it.
So congratulations again. Nice work. You seem to have done a lot of things. I find it interesting. It made a small connection with the first two presentations, because we had that discussion in the end on differences of or what would be impact of artificial substrates. So it was interesting to see your comparison and the results that you achieved. What do you think it could be the major or could be playing the major roles in the difference that you observe between the coral reefs and the inert substrates for the microfauna community?
Sorry, the major what? I didn't hear properly.
What do you think are the major players, or what are the elements that are responsible, are behind the results that you found? So which are the elements in the live coral reefs that stimulate a more dense and diverse microfauna community compared to the inert substrates?
Well, I think it's the habitat itself, because these, especially Pothylopora colonies, have a very complex hydric structure, so there is a lot of heterogeneity substrate available. So I think that might be one of the explanation.
Okay. Do you think because it's a diverse microfauna community, you have many different groups, and I can assume that there are many different species. You commented briefly about the diversity index there. Is there something else that you would think could play a role other than the structure itself, other kind of components or microorganisms or biomass associated with inert versus live substrate that could play a role?
Well, I think that, for example, the main reason that highlight the difference, especially because I think that one of the most conspicuous results was the difference in biomass with regards to the inert and live coralline. And one of the explanation is that in inert coralline substrate, most of the organisms are compressed in my case in the smallest size group, which was the 0.51 millimeter group. So although differences in densities are not found when coralline structures are compared, when you focus on this size group, you see that there are differences of how these size are distributed among both substrates, and most of these, in the case of inert coralline substrate, most of these organisms are contained in the smallest size group.
Okay, thanks. Maybe I skipped from the methodological description, but which environmental parameters did you assess, or did you assess any other environmental parameters?
Well, I didn't have the opportunity to collect the samples, but basically the two environmental parametric used was the inert and the live coralline substrate. But in case of live coralline substrate, also they were considered the colonies with different percentage of live coral tissue.
Okay.
But in my analysis, I have not considered that difference.
Okay. Do you think if you would be able to add to your analysis other environmental parameters, it could show you a similar results or not, or how much that could influence the results that you found on how related the community is to the corals? Do you think this is important? Does it make sense? My question to you. What is your opinion?
Yeah, I think it will be very interesting to consider the different percentage of live coral tissue, especially because, as I mentioned before, an important part of the community is comprised of organisms that rely on live coral tissue. So differences in this percentage might explain for example, can contribute to different in community structure, because if we found coral colonies with lower percentage of live coral tissue, it could might lead to colonies also comprised by organisms that are not obligate or facultative symbionts of these Pothylopora colonies.
Okay, I'm okay with it. Congratulations again.
Thank you, Luana. Olivia, I have a few questions. Yeah, first one is related to the. So you started looking at specimens larger than a 0.5 millimeter, as you say. I find this a very artificial boundary. And if you would take this into account and say from a methodological point of view, you go to 1 millimeter minimum or lower, for example, would you get other results?
Well, I'm not sure if I understood correctly the question, but have you said that resolution of the smallest size group is very high, like there is 0.
I know, yeah, and you have larvae as well. You have juveniles, for example, but you have also adults of certain species, which means that you miss the whole component of these adults that are so small, for example, that are potentially smaller. So my question is, do you get a correct idea on your species assemblages based on your methodology that you use?
Well, I think I did, because, for example, in case of larvae, when the larvae are considered a specific taxonomic group, so for example, we have Toea of Caridia and also Megalopa, that are larvae from the Brachyura taxonomic group. So I think that during the analysis, this difference between larvae and adult organisms have been made.
Yeah. Okay. Just a continuation on that. Did you look into these mesh sizes, or let's say the size classes, for example? Is everywhere in the world the same methodology used? Because I'm actually not really. I don't think everywhere in the world we use that kind of. So it makes it a bit difficult. That also was the basis of my question. It makes it also difficult to put it in a wider perspective, maybe.
Yeah, well, I would like to highlight that this, especially this type of research done in macrofauna communities, not considering the Corallium substrate, but considering the size group, is very scarce. So it's difficult to find literature specific for this kind of research. However, for example, this group size that I've choose are used in other type of study of macrofauna communities, not associated with coral formation, but in other types of macrofauna communities. And I think that the chosen messes I think that the chosen messes and the chosen sizes are interesting, because if you notice, there are double. So for example, you divide from 0.5 to 1 to 2 to 4 to 8. So this is why I choose these sizes.
No, okay. Another question is, could you elaborate on functional diversity?
Okay, I think
because you use the general kind of diversity indices and looking at your assemblage, but often I find these, kind of if you're a good taxonomist or a bad taxonomist, you get to other results with many of these techniques. So my question is, could you maybe go into functional diversity, because you just answered to Luana. Okay, they rely on live coral tissue, so because that would bring kind of a total, maybe another picture or kind of a special picture. Okay, these are the ones that are important, but I'm elaborating already, so maybe it's you that can. Yeah.
Okay, I will say that in this case, for example, as the organisms that rely heavily on live coral tissue are also the organisms that are all contributed to almost the whole biomass, this functional diversity could be, well, it could be altered, because if these organisms disappear due to coral mortality, the higher the contribution of organic matter to higher trophical level will be compromised, and therefore also this predator-prey interaction could be affected.
Yeah. I know you only say on predator-prey, but it was actually also on what exactly do they rely on your coral tissue?
Well, they use, well, in both, because they use coral tissue as refuge and also as food of source.
Yeah. Do they use the coral tissue as food, or
yes,
do they use the kind of associated, and then we get back actually to former presentations as well, to do they use the biofilms, especially if you go to really, so that's a bit
well, but in life are not that common. Biofilms are prebiotic are usually associated with dead coral, because the available substrate, because there is an empty substrate that is able to be colonized by other organisms. However, it should be live coral tissue. Also, these corals are also nidarians, and they have defensive strategies, so it's difficult for other organisms to colonize this live coral tissue.
And then maybe a final one, elaborating a bit on the earlier question of Luana. I had a similar question. Okay, could you, if you have to redo the study, select 3 environmental parameters that you can easily measure there? Which ones would it be?
Okay. Okay, I think I will make a differences between inner coralline substrate and not put in the same group. If, for example, the coralline substrate was covered with filamentous algae or with encrusted algae, so this.
Yeah, but can you go for biotic factors? Yeah, I would like you to select 3 that could potentially explain some things you see.
Okay, so I will choose temperature, because increasing temperature might lead to coral mortality. Well, it leads to coral mortality, and there can be an impact, especially between this difference between live and inner coralline mortality. I will also choose the suspended, because it's also related to the available food for other organic, organisms, and I will choose the and I will choose, and I think it's very important in this case, the tidal regimen, because in the Colombian Pacific this tidal regimen is quite high, so you can have differences in size of 6 meters. So I think this will have an impact, especially in live coral tissues.
Yeah, I'm glad you mentioned also the tidal regime and the wave action, because in many areas in the world, for example, you see that this is often an understudied impact on, and then you see even from same species, different sizes that occur. So that kind of so I think it's important. I think to a recommendation maybe to your the institute where you worked with to try to include this. Just to make sure that we can get one step further actually with this kind of basic research, because that's also important. Okay, that's it for me. I don't see any questions on the chat. So thank you very much for this last presentation. Thank you very much to all presenters, people that have watched, and
Thank you.
yeah, see you back for the next session of presentations in July. Of course, other students will present then, but early July we have a next series of fantastic thesis presentations. So thank you very much and good luck to everyone today. Bye-bye.
Bye. Ciao, ciao.